Would you care to come up? Hi, I'm Tim. I'm going to be chairing the talks for the epigenomics and multiomics session today for the short talks at Bioconductor 2023. First, a couple of notes for our virtual participants. Please put your questions for the speakers in the Q&A tab, and the speakers will answer them jointly at the end of all three presentations. We had 4 presentations. We're down to 3 because one of the presenters is unable to attend. So without further ado, I'd like to grant a warm welcome to our first speaker, Ryan Thompson from the Icahn Mount Sinai School of Medicine, and he's going to be presenting on forensics on multiomics data today.
Thanks, everyone. Good morning. I'm Ryan, and I'm presenting on forensics for multiomics data integration. This is a presentation about mislabeling and diagnosing and resolving mislabeling by integrating multiple omics. So first of all, what do I mean by mislabeling? It is basically any situation where the researcher believes that a data point comes from a sample that it does not come from. So mislabeling can occur in the physical contents of a sample container. You can swap 2 physical vials, for example. It can be, you can have mislabeled paper records, and you can also have mislabeled digital data files if you swap file names, for example. Mislabeling is pretty much guaranteed to happen in any large data set, and probably a bunch of small data sets too. And so you need to be aware of it, and you need to have a plan to handle it. So the first obvious problem that will happen if you ignore mislabeled data is that it messes up your statistics. So this is this plot is from a simulated RNA-seq experiment. The detail's not really important. It's just a simple case versus control. And all we're doing is introducing varying amounts of mislabeled samples, specifically sample swaps. And as you can see, looking at the red bars, as you increase the number of mislabels, your recall goes down, and eventually your precision also goes down. And the situation is even worse if you have a situation where cases and controls are always swapped with each other. And so if you can identify the mislabels and remove them, we go over to the blue bars and we see the situation is a lot better. And of course, if you can identify the mislabels and infer the correct labels and correct them, then you get the green bars where everything is fine because you fixed everything. So the point is that identifying and removing mislabels helps, and then correcting them is obviously better. Another perhaps less obvious problem when you're working with human data is problems with consent withdrawal. So in human subject research, participants can withdraw their consent for the study at any time. And when they withdraw consent, there are limitations on what you can now do with that data. I'm not going to go into the details, but for example, your IRB may require physical destruction of the samples. They may require deletion of the data. You may not be able to use that in new studies that were not in the original IRB protocol, and so on and so forth. So you need to know which samples those are so that you can abide by those limitations. And more broadly, we have an ethical, moral, and legal obligation to respect subjects' wishes regarding use of their data. And so when samples are mislabeled, the problem becomes, how do you know that you're destroying or deleting or excluding or handling the right samples to respect that consent? So this is another less obvious problem with mislabeled samples. So now we're going to talk about mislabeling in the context of multi-omic studies. So let's take this little toy example where we got multiple subjects, and from each one we take 3 blood samples. We do RNA sequencing on each of those blood samples, and we do whole genome sequencing on one of them because we don't need to sequence the genome multiple times. And so in this context, if we do a blood sample swap, then you see that the mislabeling affects both the RNA-seq and the whole genome sequencing. Whereas if you only had a swap during let's say the RNA sample prep, or the RNA extraction, for example, then that's only going to show up in the RNA sequencing and not the whole genome sequencing. This is actually a good thing when you have multiple data types here, because it allows you to infer not only which samples are mislabeled, but when in the process, at which step in the process the mislabel occurred. And this is extra important if you also generated other data from those blood samples, like ELISA, proteomics, Olink, things like that. Because if you can infer that it was actually the blood samples that were mislabeled, you can correct all of those data as well. And so the way we do this is we can make what I call a mislabel graph. So in this example, we have RNA-seq, whole genome sequencing, and genotype arrays. These all give us information about the genotype of the sample. Now we don't have the subjects. We don't have prior information about the genotype of these subjects, so we can't directly associate a sample with a specific subject. But what we can do is look if two samples have the same variants and therefore come from the same person, and we can say with high confidence whether or not two samples come from the same person. So what we can do is we can build this graph where, first of all, every sample is a node, and we start by connecting with blue edges every pair of samples that is supposed to come from the same person based on the labels. And then with orange edges, we connect all the samples that have the same genotype based on looking at the actual data. And then anywhere those two, the orange and blue, coincide, we just replace that with a green to indicate that it's concordant, that the label and the genotype are telling us the same thing. So anywhere you see remaining blue and orange edges, these are places where the labels and the genotypes disagree. And that's evidence for mislabeling. Now the node colors and shapes also mean something, don't worry about it. In 2020, we generated a data set called the Mount Sinai COVID-19 Biobank data, which is somewhere around 3,000-ish samples with genotype information, a bunch of RNA-seq, whole genome sequencing, genotype arrays, plus a bunch of other data types, like I said, that don't give you genotype information. It was collected during the 2020, the first surge in New York City. It was a very chaotic environment. And so we knew that mislabels were going to be a problem in this data set that we had to solve. And so based on this graph structure that I just showed, we built this graph for the entire data set. And then I extracted all of the connected components from that graph that have blue and orange edges, which I said were evidence of mislabeling. And I plot them here. And so with this visualization, rather than having to sift through, a 3,000 line table of potential, well, actually, no, 3,000 times 3,000, because it's every possible pair of samples you have to look at. We can look at this graph, and then the nice thing about this graph is that each connected component can be solved independently of every other component. And so by looking at this and manually inspecting each of these components one by one, the correction of the mislabeled samples took hours instead of weeks. And that was a major benefit. And so just to give you an example, this, you may have noticed that really big thing right in the center. This is that connected component. And at first it looks pretty complicated, but once you get to learn how to read these graphs, you can see that right here we have a case where 6 sample labels were actually cycled, where this one was labeled as this one was labeled as this one, all the way around the circle. And then we also see 6 other swaps, and then there's one that was swapped with a sample that we didn't get sequence data for. So you only see half of the swap, but you can still see that in the graph structure and infer what happened. And in fact, the gray samples here are samples that we didn't have data for that I included in the graph just so we could see that there was a potential swap mate for this one. And if you're eagle-eyed, you might notice that this one's also mislabeled, but we don't have anything to match it with, so it's just mislabeled. And of course, we can also go beyond just genomic information. We can use things like sample pre-processing information. We can use, like I said, those non-genomic data types. If we have a putative mislabel, we can go into the serology data or the CyTOF data and so on, and say, do we see any abnormal, biologically implausible patterns in those data types? And that'll help us confirm that there's actually a mislabel there. There's also patient information. There's an example where we had a sample swap. We weren't sure if it was a blood swap or an RNA swap, but we noticed that those 2 samples were taken on the same day in the same room from 2 different patients. So we concluded that that was probably where the swap happened in the blood sample. And over here, what we see here is I've taken all the RNA-seq samples and I've plotted them onto plates, rows, and columns, basically all the wells and all the plates where they were loaded up. And as you can see, there are some very clear patterns that indicate mislabeling that clearly happened while these are being plated. And so that tells us that those are RNA-seq mislabels. So in conclusion, identifying and discarding mislabeled samples is fairly easy, but it's suboptimal. We always prefer to correct. We have this mislabeled graph visualization that reduces a multi-week manual mislabel process to a multi-hour manual process. We can summarize those corrections as a table where you basically have one column that's the uncorrected sample ID and another column that's the corrected sample ID. And then you have your metadata table and you just do the appropriate table join to apply the corrections. I recommend labeling the original one as uncorrected sample ID so that no one uses it by mistake. Like I said, multi-omic sequencing data enables us to determine the timing and potential mechanism of mislabels, which in turn allows us to correct it upstream for their data types, and also for data generated in real time. That early diagnosis of that mislabel timing can allow us to find issues in our process and fix it for the later generation. One important lesson is randomization of samples into batches reduces the likelihood of problematic correlations in your mislabels. And I'll just leave it there. I'll just say our lab is hiring. So if this is interesting to you, please get in touch. Thank you.
Thanks for a wonderful talk. Our next presenter will be Lauren Marie Harmon from my lab, as it happens. So we'll need to switch tabs. Oh dear. We need to pull yours up. Does that look okay, Mark?
Looks good.
All right. So Lauren is a graduate student in my lab at the Van Andel Institute Graduate School, and she's working on genomic and epigenomic roots of pediatric leukemia. And today she's going to present some of our work on de-identification and re-identification, striking a balance between sample swap correction, return of results, and subject participant privacy. So hope you'll welcome Lauren, and I hope you'll find it a stimulating talk.
Thank you so much. Okay. Okay. Is this one working? No. All right, I'll use this. Okay, I'll just use this one. Hi, everyone. Thank you for having me. I'm really excited to present to you how rehash and sesame can be used to reversibly de-identify epigenomic data. So first off, what is epigenomic data? And the definition is kind of nebulous. It's anything that's above or about the genome, so anything that's not exactly the genomic data sequence. And this usually comprises things like histone profiling or DNA methylation, chromatin accessibility, or sometimes chromatin conformation capture. And this is interesting because we know that genetic information can be easily tied back to the individual, and so it needs to be protected. But with epigenetic data, it's not quite so clear as to if this can be tied back to the individual or what sort of protections that need to be in place for protection of the participants. And today I'd like to focus on DNA methylation, which is a chemical modification that occurs on cytosines and influences how genes are expressed. And specifically, I'd like to talk about DNA methylation arrays. Here we can see from recount methylation that this is increasingly popular and it's widely used. And this particular bioconductor package includes over 100,000 samples, which can be easily accessed by anyone with a bit of informatics chops to access recount methylation data. And so the question arises, this wide resource of methylation data is widely available, but is it safe for the patients that they have this data? So let's talk about what are DNA methylation arrays. First off, DNA methylation arrays are created when DNA is treated with bisulfite. This means that cytosines that have a methyl residue are protected from the bisulfite and they remain as cytosines when they're PCR amplified. However, any cytosines that don't have a methyl group are converted into a uracil by the bisulfite treatment and amplified as a T. When we measure this using infinium arrays, there are two different types of probes. An unmethylated probe that will match up with an unmethylated strand, as well as a methylated probe to match with the methylated strand. And based on how the strand is elongated, if there's fluorescence, then you can tell at a given site if it's methylated or unmethylated. And you'll note that this looks very similar to genotyping arrays. And it basically is. It's basically methylated and unmethylated is your wild type or alternative allele. These arrays also include 65 barcoding high minor allele frequency SNP probes, and these are used to detect sample swaps. So Ryan gave us really great background intro as to why detecting sample swaps is important. But here we can see methylation data from a set of triplets with 4 different tissues. And okay, it's going to be hard for me to use my laser, but on the left, you can see that the methylation clusters based on the 4 different types of tissue, as you would expect. On the right, we see the 65 SNPs, and we see that they're just straight blocks because, of course, these are triplets, so they all have the same genomic data. Of course, this is not what your data is usually going to look like because these are triplets. But if you look at this particular figure, it shows the 65 barcoding SNPs for different patients that have mixed phenotype acute leukemia. And you can cluster them by SNPs and use that to determine if there's any sample swaps. But of course, these SNPs are going to be identifiable because it's genetic information. So one easy way to resolve this is to use locality-sensitive hashing. So you call them as wild-type heterozygous or homozygous, and then based on that string, you put it through a hashing algorithm, which would come up with like a hash measurement. And using a traditional hash algorithm, that would come up with a unique string for each patient. But what we want is for it to be similar enough that you can detect sample swaps, but not so different that it's a completely different string. So that's where locality-sensitive hashing is able to resolve this issue. But what's not so easy is how we can mask off the overall DNA methylation data. There is some genetic data that is available. So for example, there are C to T transitions at methylation sites, or there's incidental measurement of extension bases. And here, this particular figure is showing that by doing unsupervised PCA clustering of methylation data from Puerto Rican versus Mexican subjects, you can see that they very clearly diverge. And note that these are 2 very similar populations. We're not comparing, for example, European or African populations here. So this just shows that methylation, there is like an inherited ancestral component to it. Furthermore, it's also hard to separate the biologically informative variation from the ancestral variation. This figure that I'm showing here from Robertson et al. Showed that DNA methylation as well as ancestry can be informative as far as cancer. So even if you look at one single gene like EDAR in the top left, panel A, you can see that the methylation profiles are different based on the ancestry. So at this point, I hope that I've convinced you guys that there is some degree of identifiableness in methylation data, and so something needs to be done about this to mitigate the risk to the patients. I guess the most conservative approach would be to put everything into controlled access repos like dbGaP. But the problem is that there's a lot of data that's already out there. The cat's already out of the bag. And even with that, there could be an issue with data leakage. Another thing that could be done to mitigate the risk is to rewrite the raw data. So at any loci that are more sensitive to privacy, it could be rewritten. And this is conceptually simple, but it's practically hard because there's a trade-off. You wouldn't want to eliminate any of your biologically relevant data. And this figure here at the right shows that with methylation, a large part of the variability is due to your biological elements, but there's also a large part that's due to your demographic variables. So here I wanted to show that Sesame already supports the rewriting of methylation files. You can use the de-identify function to rewrite the raw IDAT files. You can zero out the SNP barcode probes, but the problem with this is that it destroys the barcodes. And so the solution would be to call and hash the SNP barcodes, as I showed previously. It also has the function to do re-identification of the scrambled probes given a key. And we would like to point out that this can be integrated with public keys. And of course, the problem that this doesn't resolve is what about all of the 250,000 or so samples that are already in Geo and already available? And then the third thing that can be done is to add controlled access granular privacy. So here we can see that there's a commonly used method of encryption in computer science. So what happens is you have a plain text and it can be encrypted with a public key into a ciphertext, which can then be decrypted by anyone who has access with their private key. This is fairly general and it can be included in consent forms, and it has fewer guarantees than the first two methods, and it can also be employed in conjunction. And so this is where rehash comes into play. Here we've implemented easy to use methods for doing this. So you can use rehash to take something like a summarized experiment and to encrypt the metadata and then decrypt the metadata for anyone that has the private key. And it can also be integrated with the feature level and anonymization at the cost of some additional data duplication. And I just wanted to finish off by pointing out that return of research results to study participants is increasingly becoming a priority for us in the field. And we want to make this data available. And we really need to address the problem of the de-identification of this data, particularly before any other large companies without the best motivations do it before us. And so we'd like to point out that these tools for doing this, Rehash and Sesame, are available, but it's going to take some collaboration through the community, some discussion about how to implement this to make sure that we are doing this in a responsible way. And I'd like to finish off by thanking my lab, as well as our collaborators and our funders for our help. And thank you.
Thank you so much, Lauren. So this is of a piece with Ryan's, and I hope that people have the opportunity to consider this trade-off more as the conference goes on. Our final talk is also from my lab. So Peter Wong in my lab has been toiling away incessantly at implementing the software backend for this. And if you're grinding away at code, it can be a little bit tough to also construct a narrative. So my lab manager, Eve Gardner, has gamely stepped in to present some of the most recent developments and to agitate for partial controlled access, but ease of use for individual level variant calling from RNA-seq and whole genome sequence data. So Eve, take it away.
Thanks, Tim. Okay, so I have to first preface, like Tim mentioned, this is not necessarily my project. So any questions or mostly critiques, don't go to me. Peter will be available after the talk to answer your questions. All right, so if you're not familiar with the TricheLab already, or our fearless leader, Tim, for a long time, we've been a dry lab, as we like to call it, where we do a lot of package development and data analysis. But more recently, we transitioned to a lab that includes like some interrogation, which is actually where I do a lot of my work in the wet lab. So for today, we'll kind of talk about, does this pointer work? Okay, these bottom two kind of points. So developing packages and then using those packages to do data analysis, which in a way is how we kind of generate some hypotheses that aren't just fishing expeditions. So if we look at our latest project, we saw some unmet critical needs. As of late, I guess in 2018, the Children's Oncology Group from the NCI and the Target AML initiative published 815 pediatric leukemia patients data, RNA-seq data. And now there is RNA-seq data from more than 2,000 kids available. So triple that. And then with recent advancement in alignment algorithms, we're now able to capture the express transcriptome of these kids, which is super important and allows us to identify functional variants that are crucial for disease. So with that, we saw that there is an urgent need to officially translate this alignment data into clinically relevant genetic risk estimation. So we decided to do that. So there are some existing methods of note. The group from Lucigene in Canada attempted to do this with their package KM, which is a software used for RNA-seq investigation using k-mer decomposition. It's nice in the way that it uses targeted analysis on targeted sequences, but it requires the targeted sequences to be designed by you and requires download of FASTQs. So it's not necessarily computationally efficient, and we saw that as an issue. So with that, I'd like to introduce BAM slicing. Lauren was just up here. She was also part of this project. So BAM slicing is basically a one-stop shop for data query, count tallying, annotation, and analysis. It was built on the Genomic Data Commons provided API, which allows us to remotely slice BAM sequencing files using genomic ranges. So we utilize the API and the Genomic Data Commons bioconductor package as kind of the backbone of BAM slicing. So show you how it works. So it's a four-step process. So you query projects or experiments available via the GDC using these different functions available. And you can then download the sliced BAM files that you're interested in. So you can pick the project, the experimental type, et cetera, and then download just those specific sliced BAM files of that region, which is super great. Then step two would be to tally the reads. This was built on top of GMapper and variant tools from Bioconductor, and using the tally reads and tally variants function along with annotate with BAM info, we can use the downloaded BAM files and tally the reads and variants for annotation. As you can see, this comes out to an RDS file, which is super important later. Step three, we use the variant annotation and VEP API to annotate the variants that we just tallied using get variant annotation and get VEP annotation, along with some other functions in this realm. And then step 4 is to perform clinical outcome and genetic risk analysis. So we're able to use math tools and generate a semantic math file, which then you can use to do visualization and analysis of your data, like mutual exclusivity and co-occurring events, or as I'm showing here, survival analysis. This is a sneak preview of some data later. Okay, so I've talked to you about how we developed the package and what it uses. And I just want to quick go through 2 case studies of how we actually use the package to do some analysis from target pediatric AML data. So the first is a case study of H3K27M or I in pediatric AML. So previously H3 or histone 3 lysine 27 had been identified in pediatric high-grade glioma where the lysine was mutated to isoleucine or methionine. But recently there have been some mutations observed in AML patients as well. And so we wanted to use BAM slicing to investigate this further. So what we did was we took 11 AML-associated genes on histone 3, which came out to about 150,000 base pairs. And we sliced those BAMs from a combined cohort, these BAML and target AML cohorts, and found that through processing, we were able to go from 39 terabytes to 62 gigabytes of data, which is like, I guess your personal computer could do it a lot faster. And we were able to identify 15 AML patients that harbored the H3K27M mutation with a variant allele frequency of larger than 0.2. So we then also thought, like, let's dig a little bit further, and we looked at some other genes such as IDH2 and DNMT3A, and found in the same cohort, there are patients with co-occurring mutations from both primary and recurrent diseases, and we were able to identify them in both RNA data and whole genome sequencing data. So we proved that our method works, and you can translate from whole genome to RNA and back again with 2 different data sets, which on the same people, which is nice. So case study 2, the last one, we were able to look at some MLLT10 structural variants in pediatric AML. If you were to bulk download the BAMs, again, it would be 36 terabyte for all 2,000 patients, which is not great. So we used BAM slicing to download the MLLT1 exons, which resulted in being just 4 gigabytes for 2,000 patients. And then we were able to use our Tallying Reads and Annotate and Variant annotation functions to look within the Yeats domains, and we found these structural variants that were important. So we found, when we looked at these like Yeats domain specific mutations, we found 2 mutations, so the N115 and V114 mutations that were previously identified here in Wilms tumors, but we actually found them in our pediatric AML patients as well. So this led one of our collaborators, who's actually works with MLLT10, to look at this in mice, and they discovered that there was actually a highly penetrant and lethal myeloid leukemia model instead of Wilms tumor with one of these variants, which was really interesting. So we were able to go between organisms. So in summary, the increased ability to identify functional variants crucial for disease led us to figure out that there's a critical need to rapidly extract clinically relevant genetic risk estimation, most importantly at the epidemiological level. And before now, the existing methods fell short, speed and ease of analysis being the primary components. So we developed BAM slicing to combine functions from various bioconductor packages in order to quickly and reproducibly extract clinically relevant genetic risk estimation, which reduces computational space and time, identifies and annotates variants, and creates the ability to visualize risk. And this QR code here is for the GitHub page. So that I'd like to thank the TricheLab and especially Wally here, providing us with much motivation, and as well as our funders and our collaborators. With that, Peter will take questions.
I think potentially Eve wins in terms of finishing the earliest. 44 seconds under time somehow. I'd like to thank all of our presenters. Ryan's extraordinarily strong introduction to many topics all at the same time. Lauren pulling on the same thread, and then even Peter providing some tools that leverage a tremendous chunk of the bioconductor ecosystem, and I hope help demonstrate why this project has been so valuable to so many people. Now, Mark Carlson is my moderator to handle, or the moderator of the session to handle online questions, and I will alternate those with in-person questions if we have them. Since we have in-person presenters, I think that I'm going to request, as questions arise, the presenter come to the podium and answer them. Stop sharing. And Q&A. All right. So we have one question coming up for Ryan. I wanted to, just because in-person participants had to fly all the way here, I wonder if there's any in-person questions first, and then we'll immediately start taking online questions. So does anybody have any in-person questions? Yes, looks like we do. I think this is Nathan, if I'm not mistaken.
Yeah, my question is, I don't know if I missed it, but did you have a conclusion of like how much misannotation was happening, just like as a percentage, or I'm just like curious to know. And I realized that this was one particular time that you said was particularly chaotic. So maybe a follow-up question to that was, have you done this on other less chaotic times? And so I'm just looking for like some guidance, and like, in the middle of pandemic, human error rates are about X, and not in the middle of pandemic, error rates are about Y.
Our typical experience is that somewhere between 5 and 10% for most data sets, even the good ones. And I think that's actually about where we landed with this data set. So we did pretty good. So probably any big data set you're analyzing, probably 5 to 10% of those are mislabeled, unless they fixed it. And obviously that it matters a lot more when you're not just doing case versus control, but you've got like a bunch of different subgroups you want to look at, and some of them only have 5 people in them. Then it matters a lot more.
I think Nathan and I are both familiar with the behavior of large studies and with many hands touching samples, but certainly there's some notorious examples from Psych and Code, or even from some of the TCGA sub-projects, where there was greater than Y samples swapped. Maybe the most notorious example, oddly enough, is from genome assemblies, where 8 out of the 9 original orangutan assemblies were actually swapped, and had to be corrected. The authors finished their article with a coda. This is from the WashU Genome Center. The authors finished with a coda of, we encourage all further studies to please cite both of these and note the swapped sample labels. There was a newly discovered species that had been mislabeled as an orangutan, for example. So I claim that this is quite pervasive, and it's exciting that there may be better ways to cross-check. So I think our online question is also for Ryan. So if you wanted to take that, choose
Sure.
your mic.
Yeah. So the question is how long it takes to construct the graphs, basically. First of all, I want to say the speed up is in how much manual annotation effort is required, not the computational time. Constructing that graph doesn't take long at all. You just have to load in the RNA-seq counts, or no, not the counts. No, it takes a while to run the tool called NGS Checkmate that does the genotype matching. It takes a little while, but not that long, because it's a k-mer-based approach, so it's pretty fast. It's like you can run that whole data set of 3,000 samples in a day on a cluster. And then you just have to look at the graphs and solve them one by one. And once you learn how to read them, it's pretty fast to solve each one, even that big one that I showed.
I have a follow-up question. A lot of graph algorithms in CS are notoriously NP hard. Is this something that can be reduced to or heuristically solved as click or
I have
Or just connected.
tried to write a heuristic to solve it. It solves only the simplest of cases, and it's like 2 pages of if-else cascades. So we've actually hired a person who's going to be working on packaging this and also working on maybe an algorithm that's a little smarter than a giant if-else cascade.
I think program officers often accept more research is needed as an answer to that one. Do we have further questions in person or online for our speakers today?
Could you say, if it can be answered, could someone say a word or two about the remote infrastructure for BAM slicing, what the requirements are, how the tooling works?
Yeah, that's you.
Can you say? Can you repeat the question?
So the question
You
is,
should take the podium. You should get
okay, now we're all in position. So for BAM slicing, presumably there's some remote computational structure that's in the vicinity of the large BAM files. What does that infrastructure look like? What's the tooling? How hard would it be for one of us to implement this BAM slicing approach in our lab?
So I guess the most challenging part is already handled by the genomic data common portal. Like they already established these infrastructures as a BAM slicing API that's allowing people to remotely slice the regions of interest of those BAMs or harmonize and store on the cloud. Yeah.
I'm going to actually extend this question a little bit further because it raises another one. The pulling coordinates or ranges of index coordinates from BAMs is easy. And we have a keynote from Hung Lee. One of the great features of SAM and BAM is the provision of an index to do that. But maintaining controlled access is hard. And so would it be fair to say that this problem reduces more to one of authentication and authorization than of storage or rapid indexed access? We glossed over the GDC tokens, but that's actually a critical feature of the genomic data commons that's not necessarily a computational difficulty so much as an infrastructure difficulty.
Yep.
Would you say that that's fair, having worked extensively with GDC tokens, expiration, and essentially finding out firsthand the added complication of maintaining controlled access in a way that expires? Is that a fair statement?
Yeah, I think it will benefit the community if it's not access control, like allow people to quickly explore the genetic information stored in these bands.
But so if we do that, then we have the issue that Lauren mentioned, which is that separating the genetic identifiable information from biological or condition-specific information becomes quite a chore. I'm not saying that I or we have a solution, but I'm hoping that people will cast it in this light, because as the Recount 3 project showed, for example, even RNA-seq counts are turning out to be re-identifiable. So this might raise this might be a time of reckoning for all of us that work on large-scale genomics data.
Yeah, I also, I agree that, yeah.
Well, fair enough. I guess we're all in agreement then. Do we have further questions for the speakers? Nathan, you're smiling endlessly. You sure you don't want to lob one? Ryan? Yeah. All right. Well, thank you everybody for attending, and I believe lunch is next. And if you want to give another warm round of applause for our speakers, we have time for it. So after that, off to lunch.